Okay. So why to deal with the biomass or the size? Because it could be as a primer indicator related to substrate characteristics, also some phenology indicating the thermal changes. And it could be also the supporting parameters for the abundance, because the most of indicators in Water Framework Directive is based on the abundance, and it would be nice, for example, for food webs or feeding strategies to involve also the size of the animals. And something wrong with the movement between the slides. Yeah, I'm maybe sure how I do it. Yeah, I think now. Okay, thanks. So this is the research question: why to involve this biomass? Because it could be better for the functional parameters of ecosystem, like decomposition or nutrient cycling. So we generated, or we compiled the first version of morphotaxa of these macroinvertebrates. There are around 100 morphotaxa, and the basic characteristics of these morphotaxa was that they are uniform for the modeling relationship between the length and the biomass. So this is the picture of these. So we are starting with the scale image of specimen, of individual specimen, and make a classification, identification to a level of morphotaxa or more detail. Image is editing and thresholding to get a binary file for the evaluation of area of the image of the object. And then we measured the length and area of the animal. In the first stage, which was calibration, we use the manual calibration, manual measurement, but there is a good opportunity to involve part of these processes to be automated or AI trained. So in the final, we have some data for biomass estimation and so also size spectrum of community, and for certain species also the instar identification as relevant for my chironomids, which I'm studying. So we have two scenarios. First one is with the measurement of the length, and we have the opportunity to get some equations from the literature review, how to transfer the length of the animal to the biomass. And the second scenario is based on measurement of area. Then we recalculate it to the length, and again, the remain of process is the same. And within this part, we have the space for the calibration, for the exploring the relationship between the area and the length. So this is only visualization of our process, measuring the length and the image area and using image analysis software. So we have almost 2,000 labeled images with the scale using for this calibration. And together with Jurat, we are working on some automated recognition of this morphotaxa. And maybe we will be also able, with your help, to automate also thresholding and the measurements. So the challenges are automate recognition of some structures. For example, for chironomids, it's not only important to have the entire body area, but also to measure only head capsule. So if some automate procedure would be able to identify the head capsule and measure it, it would be fine. And the second challenge is to measure the picture, which has more than one specimen. So this is the table with the parameters collected from the literature about the relationship between the length and the biomass. So you can see the type of the relationship and some indices, and these are the taxa. So results. I selected 4 examples for 3 minutes, I see. Chironomids, the length measured, you can see ranges, length calculated, the type of the relationship is not final, we will test it. But in this case, logarithmic relationship between the area and the length, so you can calculate the length and substitute to the formula for the calculation of the biomass. Same for other groups, I will skip it. And finally, for Hydropsychidae, you can see the body area from the image and the dry weight biomass calculated calculated from these formulas. Some challenges: automate recognition. Some success made Durat with his team about the recognition. So we are working on training of this model, but we have a problem with the selection of subject because if the background is not very uniform, so there are problems. A little bit surprisingly, for Photoshop was the most efficient in the separating of objects from the background, but we can use also some other software. And so I will see, this was the middle way approach that we found. There are species traits about the maximum size of the body. So we used for the calculation of proportion of individual feeding strategies like this one. And with this question mark is when we finish from the case study measurement of individual size, individual specimens, so we can generate such a picture also for the real pictures or for the sites within this use case. So in summary, we have these images for calibration. We have additional 5,000 images for the training and for the evaluation. And there are some future developments related also to taxonomy, instar separation, and others. But the main thing is to thanks to all colleagues which participating on sampling, sorting, imaging, and image analysis, also lab imaging, and Elbe River Basin Authority providing the implementation to the practice. And if you have some solution for question and challenges, I will be happy to hear it. So thank you very much for your listening.
Thank you very much. We have time for questions. Yeah, Gordon?
You mentioned the importance of having the examples of Leadstone doing the calculations. Do you ever get a case, I know in say mammals and others can happen, where you get one species that's like a smaller or a scaled up version of another? There was an infamous case of supposedly a wild panther on the loose of some wild some Moorlandian remote part of England. It turned out it was a domestic cat, just photographed close up. Now, I don't know if you're getting that extreme, but could that be a potential problem where the example images don't cover examples of a species that's either scaled up or scaled down version of the examples that do come?
I'm not sure if I understand.
Oh, sorry, too fast. Do you ever get one species that the length scale calibration breaks down because the other species is either a bigger or a smaller version, but otherwise similar looking to the other species?
Yeah, but the images are scaled. Also, we are comparing, if you mean the individual stars or you say, working like stars or
The different species and different size scale.
I think that we are working with the definition of morphotaxa. We try to use is usually the families. So within the RC, so within the family, you can find the relatively similar life cycles. So there is not in nature of